Anna Murphy, our Product Quality Manager here at Horizon, will be presenting on the Blueprint Blue Sky proposal. Hannah is a medical biochemist turned quality professional with a strong interest in the quality and regulatory frameworks. And our other presenter, I'm very pleased to announce at Visiopharm, is Martin Christensen. He is the Director of Sales and Products Manager at Visiopharm, who many of you may well know. Martin has a strong interest in digital pathology and continuously supports pathology laboratories in the transition to digital pathology. So without further ado, I'd like to pass you over to your very first presenter of the day, Hannah Murphy.
Thank you, Joe. Hi, everyone. It is a pleasure to be presenting this webinar today. This webinar will cover recent developments, tools, and research in the field of immunohistochemistry. Much of what I will explore will be based on the FDA AACR ASCO public workshop, as well as developments within Horizon Diagnostics. So to begin with my first slide, covering the impacts of biomarkers. In the 1990s, the framework for companion diagnostics was much more of a one drug, one assay paradigm, with around 5% of drugs being based on a targeted therapy. As we move into the 2013, we can see that it moves to more of a 45% targeted therapy with multiple drugs and multiple assays. If we look back to the 1990s, it was one drug, one disease indication, one test, and one allele. For example, the Abbott break-apart fish probe for the drug crizotinib. As we now look into the present time, we move into EGFR with the two different assays. So we have the Roche Cobas and the QIAGEN TheraScreen, each with their own individual drug indication. So new biomarkers are clearly increasing in complexity. There are different tumor indications and different combinations of influencing biomarkers. There are different IHC antibody clones available. There are different staining protocols and different platforms. There's different clinical decision points and different assessment methods. There are also challenges in the nature of the assessment. So there's challenges faced by the limitations in IHC precision and challenges by the limited biopsy tissue, particularly for small cell lung cancer. So these challenges exist within the clinical pipeline, and there are several opportunities here with new tools and technologies can assist laboratories with the development and routine monitoring via internal controls. If we look to the beginning of the slide here, in research and development, whether that be assay development, sample screening, patient stratification, there's challenges in the availability of clinical samples. Rating the tissue archives presents its own difficulties, especially for those rare variants. As we move into the second part of this slide, the clinical studies, the different phases of clinical trials, the clear evaluation, and patient stratification, these all present new clinical challenges. So in clinical trials, you have multiple partners, multiple geographies, and these can lead in a drift into assay performance. Outsourcing to clinical research organizations can make managing variation much more difficult. There's also unnecessary and sustained variability that can affect study performance. And then to the final part, on market companion diagnostics and proficiency testing, there are a number of other challenges here, such as manual evaluation methods, leading to the potential for variation. New drugs rely on successful and effective adoption of diagnostics to ensure their reimbursement. And finally, proficiency testing, ensuring that standardization and accuracy of diagnostic testing are met. So the regulatory challenge is quite significant. There's new biomarker complexities that impact this. The safety and efficacy of new drugs has a large emphasis on marker positive, without always necessarily considering the impact of those treatments on marker negative patients. There's multiple tests that have the potential to change the selected group for the test. So just because one variant or one biomarker is indicated by one assay and another one, it doesn't mean that those two tests are necessarily equivalent. For new tests, for new drug treatments, bridging studies may become necessary to support safety and efficacy. And there's the potential, as we go more and more away from the one drug, one assay, into a matrix of companion diagnostics, that there's a real risk of mismatched approved drug and device combinations, yet alone without even considering the LDT scenario. So to put this into context, at the FDA AACR ASCO public workshop in March earlier this year, there was a large discussion that took place with regard to the particular biomarker Pdl One. So what is Pdl One? It's a biomarker with a major role in suppressing anti-tumor immunity. It shows variation in tumor expression and response. It's subject to a range of pre-analytical and analytical variables, and shows differences in application of assays. Therefore, it will become more and more important to define stratification for patients. Several open questions even still remain on the utility of Pdl One as a predictive marker, especially in comparison to other biomarkers. So the PDL challenge, just to put it into a few summary words here, there's 4 to 8 drugs in development, each with their own parallel development programs, each with variation in clinical trial design. And in addition to all of this, there's multiple companion diagnostics. There is a different test for each drug. So this is the introduction to the blueprint and the blue sky. So the blueprint proposal is derived from the work undertaken by an industry consortium that was proposed and discussed at the FDA AACR ASCO public workshop. Blue Sky is in the development of novel IHC tools, and in this discussion we will focus on Horizon Diagnostics and Visiopharm. So in terms of Blue Sky development, this year we launched at Horizon Diagnostics a range of IHC reference standards, and we're continuing to increase that range in developing new targets such as Pdl One. So the advantage of our reference standards are that they provide an unlimited supply mechanism. They are cell line derived and therefore providing consistency and accessibility compared to tissue archives. We undergo a reproducible production pathway, taking the cell lines right the way through to the on-site controls. It's independent from tissue archives, and so therefore can be used as a supplement and as an addition. The same material source, being the cell lines, can be used for multiple tests. You no longer have the issue of availability of tumor material for validation. The same material can be used for NGS testing, for IHC testing, or for FISH testing. And they provide an on-site control. So every slide is like a miniature assay. It has its own environment, its own staining, and therefore the on-site control provides a direct reference point to the material that may be placed on that slide. And in our development, it was critical to us to find a reproducible method of QCing our material, and we undergo on a series of assessments using quantitative digital pathology. And it's at this point I would like to hand you over to Martin, who will further explore the value of quantitative digital pathology.
Well, thank you very much, Hannah. So, as Hannah mentioned, tissue-based immunohistochemistry data is already used extensively in clinical trials, and in some indication areas as drug treatments, this type of input may increase further in importance, as we've already talked about. So the only problem is that there is a huge variability between centers in staining protocols, staining quality, and even in the reading and interpretation of biomarker data. And all of these can have a great impact on both precision and accuracy of data. So at a very fundamental level, the challenge we're facing in all of these situations is data quality in the sense of sensitivity, specificity, and reproducibility. And when it comes to tissue-based data, the journey from biopsy to data is very, very long, complex, and with several challenges along the way, So it's not difficult to imagine that any one of these steps can cause some artifact or some errors or some variations. So some of these steps include tissue preparation, which also includes fixation, tissue thickness, and all sorts of other things. It includes staining protocols, staining sufficiency, and of course the last, the reading, interpretation, and quantification of the biomarker expression. Now, you've already heard from Hannah that some of these challenges can actually be solved quite efficiently using genetically defined reference material and cell lines, but it's also well known that manual assessment of biomarker expression is associated with quite a big significant inter- and intra-reader variability. And in some cases, there are also limitations when it comes to sensitivity and specificity of manual biomarker assessment. Now, when we start to fill out some of these holes and apply standardization, we get closer to a complete standardized workflow of working with human samples and also with biomarker development. So let me just show you one of the difficulties in reading and interpretation when it comes to biomarker expression. So these few series of images illustrate my point quite well. So here we've arranged a Ki67 stained nuclei sample from lowest reacting to highest reacting. And then we've asked just one pathologist in this case to determine at what point she would call a nuclei positive and negative. And as you can see, many of the ones to the right are all negative, and you can see the nuclei in question is highlighted with this red circle right next to it, or red square, sorry. And we have to go to the second column to find the cutoff point between negative and positive. But in my non-pathologist opinion, but just as an image analysis expert, there's a very, very little difference between the negative and the positive nuclei, and it can be extremely difficult as a human to consistently detect that minute difference. And of course, I have the luxury of visiting a lot of pathology labs, research places, and industries, and it's very, very rare that I have two pathologists or even two people setting the exact same cutoff. So some would believe that the top image to the right should also be positive. Others would believe that the positive boundary should be a bit lower in that setting. So it's very, very difficult to standardize the reading and interpretation of these biomarker expressions. Now, in multiple locations, we have shown that image analysis can be used, if used correctly, to effectively improve both the accuracy and the reproducibility of biomarker assessment. So here I've included just two examples, but of course there exist many more. So in the one to the left, the pure contribution of inter-reader variability associated with Ki67 assessment was quantified across 20 tumors and 126 participating labs. So this was a huge study conducted by the Nordic QC. And in that study, it was demonstrated how image analysis can be used to significantly reduce inter-reader variability by using a different method to quantify the tumor areas and then quantify the positive and negative nuclei. So in another study, the one to the right, which was part of the National Danish Validation Study for HER2 assessment using algorithms, it was demonstrated how improved sensitivity and specificity of the algorithm when quantifying HER2 protein expression could significantly reduce inconclusive cases when comparing to gene amplification. Now, when reducing the number of inconclusive cases, that translates directly into huge time and cost savings, especially because you can limit the number of reflex testings, which typically require more expensive biomarkers such as HER2 FISH. Now, there's a lot more detail offered on our website in our other webinars on these kinds of studies. So, of course, we see many obvious applications for this kind of automated method using image analysis. So by automating aspects of stain quality control, it would be possible and maybe even scalable for EQA organizations to offer more frequent, perhaps on-demand proficiency testing and calibration services, maybe even using genetically defined material together with image analysis to standardize as much of the process as possible. it will also be possible to perform an objective and quantitative standard estimation to improve the compliance and the protocol recommendation given by these EQA organizations. And in multi-center clinical trials, it would be a lot easier to standardize and monitor the data from each of the centers because they can all use the same method, the same reference material, and the same reading algorithms. And of course, it's our big hope that larger diagnostic pathology labs would be able to benefit from such methods to closely monitor drift in staining quality for biomarkers, but also use the same kind of algorithms on their human samples for patient diagnostics. So with those few words, I'll leave it back to you, Hannah.
Thank you, Martin. So to continue on from that, we're looking more now at the Horizon Diagnostics Blue Sky application. So I'd just like to discuss a number of key questions. So the first question, does your assay accurately and reproducibly measure what you expect it to, what you say it does? Does the assay actually identify a biological difference of clinical significance? Does your validation show evidence to support improvements in the clinical workflow? And how do you monitor for your IHC analytical performance? So those kind of questions, if you just have them in the back of your mind as we continue through a case study with one of our partner laboratories. So the IHC Blue Sky application can be shown by a number of utilities of the reference standards. The performance can be assessed when optimizing a protocol or platform. The reference standard can guide the evaluation of antibodies. Protocols can be assessed with the use of the same antibody clone, and concordance or discordance can be assessed to determine reproducibility. As part of our development of an upcoming variant, the Pdl One IHC reference standard, we've produced an IHC Pdl One array containing 8 cell lines with negative and positive protein expressing levels. The results shown here show the Pdl One expression levels using the cell signaling antibody shown at the bottom here. Here you can see the positive in the brown stain and the negative cores in blue. To improve our confidence, a part of our assessment was to use quantitative digital pathology, and these two charts show our initial evaluations showing the percentage positive and the H scores for each of those individual cores, allowing us to see a clear difference between what is determined to be positive and what is determined to be negative. And here you can see the individual images of the positive reference standard and then the negative. In working up our development, we provided our reference standards to a top US reference laboratory with a validated protocol for Pdl One, and this was their results in comparison. As you can see, that this antibody used the same antibody at the same concentration, and we can see here that there is a lot more positive staining even within the negative cores. And here we have the close-up of the positive core and then what is supposed to be the negative core. And you can see a large amount of off-target staining, and it clearly there needs to be some investigation that takes place here to understand why the negative core is presenting as positive. And the laboratory did some investigation and performed an orthogonal method that proved actually, in fact, on the basis of RNA, that there was an issue with the assay and not with the performance of the reference standard. So I just would like you to think about how you monitor for your everyday reagent performance. How do you evaluate potential fault points, such as the serial dilution of antibodies, and consider how LDTs both have positive and negative aspects, but how this potential drawback could be mitigated by using new technology. And finally, I would like to touch on the great work that undertaken by the IHC Blueprint proposal. The goal of this group is to package information together on analytical comparisons of various diagnostic assays that may be conducted. This includes understanding the analytical performance of different Pdl1 biomarker assays. The studies are being The samples being used for this study are a mix of sample types that are representative of target patient populations, with a focus on non-small cell lung cancer and Pdl1 testing. The staining will take place on investigational use only assays at the individual diagnostic stakeholders, and the evaluation will take place both by a company pathologist and an independent third party. I think this kind of study goes to show what great research is being undertaken, and Horizon is always a great advocate of these kind of studies, so we're very much looking forward to seeing the results. So to the penultimate slide, this just shows Horizon Discovery's range of different products and services. For us, collaboration is absolutely key, and so we engage with a range of different industry stakeholders and academics to provide an end-to-end product and service to support changes in healthcare practices.